All right, good morning, everyone. Thank you for spending your rainy Friday morning with the Adirondack Park Invasive Plant Program. My name is Tamara Van Ryn, and I am the manager of the program. And I am delighted today to be joined by Charlotte Malmborg from the New York State Hemlock Initiative. I also have on the phone Zach Simic, who is the Conservation and GIS Analyst for the Adirondack Park Invasive Plant Program. And Zach's going to help out today on this webinar. Before I get started, I'm really glad that you're all here. I wish that we could have been at Adirondack Lodge with all of you last Sunday doing this out in the field, but so glad that we could have that Charlotte was able to join us and we could do it via webinar. I think all of you know hemlocks are such an important part of our landscape in the Adirondacks, and it's wonderful to have all the interest that we have today in learning more about hemlock woolly adelgid. So I want to thank today not only Charlotte for making up for being able to do this as a webinar. I also want to thank the Adirondack Mountain Club, who was our co-host for this program last weekend and who's done a lot of great outreach, and the members of the Mountain Club who are out there in the woods every day, who are great eyes and ears on the ground. Today, I'm going to go over a few features of Zoom, just so you know how this will work, and then I'm going to turn it over to Charlotte fairly quickly. So you can see here the number if you're having trouble with dialing in on your computer. A couple of things, just a couple of hints on Zoom. You can mute and unmute your line and start video. If we hear a lot of background noise, we will actually go back and mute you because we want to make sure that we can hear Charlotte clearly. You can also start your video if you need to, but just in respect of everybody who's working on slow bandwidths, we're trying to keep the video down a little bit today. And also, if you use those up-down arrows there, that will help you know if you're not getting good sound or you're not getting good video. You'll get some drop-down menus there. Right there, you can select your microphone, you can select your audio settings here and your video settings there. Just also wanted to point out that if you go to the Manage Participants button, you can raise your hand. Zach's going to be helping out, making sure if we see your hand raised there, you can also mute and unmute from that slide. You can also go down to the chat function on the bottom of your screen. We will be monitoring chat today. In the chat function, you can send a message to everyone. That's all 50 or so people who are on the call. You can also just send a message to one individual. So you could do that, or you could just select one individual. So with that, I'm going to actually stop sharing my screen, and that's going to enable Charlotte to start sharing her screen and get going. So Charlotte, while you pull that up, I'm just going to thank Charlotte for adapting to a webinar again. And Charlotte is the Education and Outreach Coordinator for the New York State Hemlock Initiative. And in addition to hosting education programs, she helps manage a whole network of citizen science volunteers. And with that, Charlotte, I'm going to turn it over to you.
Awesome. I hope everyone can hear me.
I can hear you great, so that's working good.
I was going to say, of course, if people can't hear me, they can't answer, so it kind of defeats the purpose. But I want to thank everybody again for joining us. So today I'm going to give a presentation. I've called it Conserving the Foundation, Keeping Hemlock Trees in Adirondack Forest, because that's what we're really doing when we're conserving hemlock trees. And I'm going to talk a little bit about hemlocks in the beginning, but then I'm going to move and talk about the threat that is posed to hemlock trees in our forests today, the hemlock woolly adelgid, and then I will go on and we'll talk about management and what you can do to help. So, it appears that my presentation isn't moving. Oh, there we go. Hi. So I'm Charlotte Malmborg. I'm the Education Outreach Coordinator for the New York State Hemlock Initiative, which is part of Cornell University's College of Agriculture and Life Science. And today, as you now know, I'm doing this in partnership with the Adirondack Prism, but I also have to mention our supporters, the New York State Hemlock Initiative supporters, including the New York Department of Environmental Conservation, the Forest Service, USDA, APHIS, and the United States Environmental Protection Agency. So I'm going to get rolling today by starting to talk to you about hemlock trees. And we're going to start with identification so that you all know and can go out into the field after we're done and socially distance yourself from others and get up close and personal with the trees. So a few of the key identifiable features that I'll go through. I'm going to start by talking about the bark. And bark of hemlock trees is typically brown. It's rough and furrowed as well. So it's got these kind of scaly features going up, especially as it gets very large. Hemlock trees can live up to 400 years and beyond that. And they also grow to be rather tall. They can be about 175 feet tall when they're mature. And another thing that they get when they're mature, as those furrows start to appear on the bark as the tree ages, you might see a reddish, the reddish inner bark coming out. So that's a really good key identifiable feature of hemlock trees. I'll talk next about the foliage. They are evergreen conifers, so you're going to have that needle foliage, not necessarily leaves, and they have kind of a lacy, airy texture. Very feathery in appearance, as you can see from that background photo that I've used. And they're evergreen, like I said, so you can see those needles all year long. This is a great time to be looking for hemlocks because you're not going to see any other trees with leaves on. So a lot of the green that you see in our forests right now is hemlock. Kind of one of the best key identifiable features are hemlock needles. They're very different from a lot of other conifer needles. The first thing is that they're short and flat. So they have really short needles, like pines have much longer needles. And spruce and pines both have needles that are round that you can roll between your fingers. Hemlocks, with their needles, you can't do that. They're flat. So if you try to roll them between your fingers, they won't roll. They're about 3/4 of an inch long, so they are quite small, and that contributes to the airy, kind of feathery appearance that they have. On the top, they're very shiny, and they have a deep dark green color. They've got rounded tips, not pointed tips, as you'd see on, say, a spruce tree. And they've got an opposite arrangement. This is a really important key identifiable feature of hemlock trees, that their needles are arranged flat on the twig opposite each other and not whirled around like you see on some other. And another really great way to identify hemlock is to look for the two distinct white stripes that they have underneath their needles. Their cones, when their cones are out, are small. They've got these nice rounded scales, so they're about 3/4 of an inch to an inch long. They're very tiny little cones. Got those rounded scales, and they're brown when they're mature. When they're immature, they kind of look like little green footballs. So next I'm going to talk a little bit about the distribution and density of hemlock trees in our forests. And if you take a look at this map here, you'll see that hemlocks are widely abundant throughout New York. Even those areas that seem to have spotty or no hemlocks actually have quite a few. It's a very common tree in our forest. But the densest regions of hemlocks, the first is the Adirondacks. You guys have the densest hemlock stands in New York. So it's a really important area for this tree. The Tug Hill Plateau is also a really dense hemlock region, and the Catskills are probably the third most dense region of hemlocks. Where I live in the Finger Lakes, we have a lot of the hemlocks lining our gorges that kind of give the really distinct landscape that the Finger Lakes have. So they're a really important tree, very common in New York. They're actually the third most common tree in New York. I always get asked what the first two are, and I'm pretty sure they're sugar maple and maybe silver maple or red maple, I think. So sugar and red. So those are the first two. But hemlocks are the third most common tree in our forest. So they're a really, really abundant tree species. And ultimately, because they're so common, because they make up a lot of our forests, and because they provide a lot of different and unique habitat structures in our forests, they're what we call a foundation species. They really create the forest ecosystem that they live in. So these habitat building blocks that hemlock trees are, they do this in a bunch of different ways. And so I'm going to go through kind of 4 major distinct ways that hemlock trees create the habitat that they live in. The first is that they're filling a really specific niche. So hemlock trees often grow in a lot of places that other species do not. And most specifically, this includes steep slopes and shady areas where a lot of other tree species aren't able to grow and thrive. They also help support an entire food web. They're a really common food species in the winter for deer and porcupines. They also support over 400 different forest canopy arthropod species. So that's insects and spiders, creepy crawlies. So between migratory birds, the mammals, all those little creepy crawly bugs that are obligate to hemlock, you're talking over 400 different forest species rely on hemlock trees. Hemlocks also provide unique ecosystem services in our forests. The most important key ecosystem service that they provide is help keep freshwater streams cold and clean. For temperature, they provide direct shade. They're very shade tolerant, but they also provide a lot of shade with their dense canopies. And that keeps water cold and creates these little pockets and microcosm habitats that really rely on cold water systems. And they help keep water clean. They have a really shallow branching root system that naturally filters water as it approaches our streams. So they're a really key species for providing freshwater stream conditions that are important for life and for us. I mean, we also love drinking fresh, clean water. And they create these unique habitats. So in addition to the temperature regimes, they also create really distinctive soil and water chemistry conditions. And so they're really important for species like brook trout. So any trout fishermen on the line should thank their hemlocks for keeping the trout habitats alive and well in New York. But they also create unique soil conditions because they have really tannic properties. They make things slightly more acidic. And this creates these unique habitats and improves the overall biodiversity of our forest. And so they're a really important species for keeping our forest ecosystem stable and diverse. So hopefully I've convinced you that hemlock trees are very important in our landscape, worth saving. But another thing is that they are really just a part of us. They're part of the natural fabric of New York's forests. And when I talk about New York's forests, I'm talking about 19 million acres of land is forested in New York. It's quite a bit. And I'm really talking about how we can be better stewards to our forests. So recreators, hikers, bikers, skiers, anglers, boaters, we all play a really important role in forest stewardship. So recreators play a really large role. And I want to talk specifically about landowners as well. Landowners play a really, really outsized role in forest stewardship because about 76% of New York's forested land is privately owned. So landowners are a really huge, important part of keeping our forests healthy. So I'm going to talk about the hemlock woolly adelgid, which is the threat facing our native hemlock trees in New York. But first I want to just give you an idea of what our forest would look like if we lose our hemlocks. And this is a photo from the Pisgah National Forest in North Carolina. The Southern Appalachians have been hammered really hard by hemlock woolly adelgid. And losing hemlocks, especially in such a hemlock-rich area, looks something like this. And this is something that we really don't want our forest to look like. And certainly, you know, we're losing not just a beautiful forest tree, one of the prettiest trees in our forest, we're also losing all those important ecosystem services that I just talked about. And so we really don't want to live in a forest that looks like this. So now I'm going to talk about the hemlock woolly adelgid. I can check, are there any questions about hemlock trees in general? Okay, so I'm going to move right on and talk about the hemlock.
And Charlotte, I'll just add, if somebody's asking any questions, we will keep monitoring the chat there. So please go right ahead and add any questions you have into the chat if you have questions for Charlotte as she goes, and we'll make sure that we stop and pause and let Charlotte know she's got questions. All right, go ahead, Charlotte.
Great, thank you very much. Okay, so now I'm going to talk about the Hemlock woolly adelgid, which is this insect that has invaded a lot of areas of New York's forest. It is an invasive hemlock pest, hemlock specific pest. And I'm going to start by kind of framing this, the invasion of Hemlock woolly adelgid, because Hemlock woolly adelgid is not necessarily an invasive pest everywhere. There are many species of hemlock worldwide, most of which are in Asia, but we also have 2 species of hemlock trees in the Pacific Northwest and 2 on the East Coast of the United States. And Hemlock woolly adelgid is actually native in a couple of these ranges. And that includes all of the Asian species and all the Pacific Northwest species. So Hemlock woolly adelgid is a native pest in these other regions. And so in these places, they have natural predator regimes in place, managing populations below widespread hemlock mortality. So there is a natural system in place that does take care of HWA where it lives. However, the invasive range where HWA is a non-native species. And that includes our native hemlock, Tsuga canadensis, and Tsuga caroliniana, which is a native hemlock species in the southern Appalachians. So this is what a Hemlock woolly adelgid looks like if you put it under a microscope. You've got this kind of round beach ball-like body with a piercing, sucking, straw-like mouth part. And so you've got this kind of coiling mouth part. It's all kind of rolled up there, you can see. And it's also got these pores along its back, and that's where the wool is excreted from as it feeds. And so HWA it is one species that infests all these different species of hemlock tree, but after a while, those populations have separated into distinct biotypes that are kind of genetically linked to the region that they live in. And what we found is that the hemlock pest, the Hemlock woolly adelgid biotype that we have here on the East Coast is actually native to southern Japan. It's closely related to the populations that are from that region. And that's based on work that was done by Dr. Nathan Havel with the US Forest Service. He discovered that the southern Japanese HWA has very similar genetics to the HWA that we have here on the East Coast. So that's where kind of our populations originated from. And one of the ways that we think HWA may have been introduced to the eastern United States is infested nursery stock. And so that the fact that the HWA that we have here is most closely related to southern Japanese populations kind of indicates that it was brought here from that region and possibly on infested nursery stock. So I'm going to talk a little bit about HWA spread in New York. So the original populations of HWA arose, it's the first HWA infestations we knew of on the East Coast were found in Richmond, Virginia, and it quickly spread kind of north, south, and west from that area throughout the southern Appalachians very quickly, and it's been slowly making its way north towards New York. But the first infestations in New York actually came through New York City and Long Island, which is another reason why we think infested nursery stock is a common vector for HWA. That's an area where there's a lot of trade and a lot of development. So we know that trees are kind of going through that region a lot, getting shipped other places. And so the 1980s is when we kind of got our first HWA infestations. And it quickly moved north, so from that origin point, by the late 1980s, it had reached the Catskills, so it went up north into the lower Hudson region and into the Catskills. We got our first infestations in the Finger Lakes in 2008. Those were when the first ones in the Finger Lakes were detected. And it's spread north into the Southern Capital region as well. And it's been found as far north as Ballston Spa. But I am going to talk about one particular infestation that occurred that's no longer on this map because it was treated. And that's in 2017, there was an infestation found in Warren County. We were really, really lucky that the guy who found it just happened to be an HWA researcher himself, Dr. David Orwig, who is at the Harvard Forest in Massachusetts. He studies HWA. He found it in the Adirondacks and he immediately reported it. So he found it in July, and they were able to treat this area, to survey it very, very closely and treat that area in October. And what that story shows, and that's a photo of the actual infestation on Warren County. And what this story shows is the importance of early detection. So that infestation was quite small, I think, and Zach can correct me here, I think there were 3 trees that had HWA that they found after they found that first infestation. And ultimately, all those trees were treated, and they were able to beat that population back. And that early detection really led to a rapid response. And what that rapid response does is it helps slow the spread in that region. So we now know that although HWA has been found in the Adirondacks, they were able to respond really quickly to those infestations. The trees were treated, and now, as of now, we don't know if there are any other Adirondack infestations. Once again, Zach can correct me if that's still true. So a little bit more about the Hemlock woolly adelgid. We're going to talk next about the identification and some biology, including the life cycle of HWA. So the best way to find HWA is to look for those white, waxy, woolly masses that appear on hemlock twigs near the base of needles. And HWA, as I said, has that piercing, sucking mouth part, so they're feeding directly from the twig tissue, and that's why you're going to find them near the base of needles, directly on the twig, and not on the needles themselves. So as I said, they're feeding directly from that twig tissue, and that feeding action where they pierce into the twig tissue does cause a lot of damage. Ultimately, when you get a population density that's high enough, the flow of water and nutrients is blocked. So the tree can no longer put on any new growth, and eventually the tree dies. And it takes about 4 to 20 years to do this. Usually about 4 to 10 in the southern Appalachians, where you're not getting cold winters knocking populations back. And up here, the timeline is a little closer to 6 to 20 years. However, with a trend towards milder winters, we may see creeping closer to the 4 to 10 year range. So now I'm going to talk about the life cycle and the—
Actually, Charlotte, I'm going to interrupt for just a quick question. This is Tamara, and I got a question sent to me privately that fits into your description of how you identify it. But someone asked when is the wool seen? Is it seasonal? Is there a better time of year to look for the wool? Can you answer that one?
So I'm going to say no spoilers. I'm going to talk a little bit about that when I talk about the life cycle, because it is tied very closely to the phenology of HWA.
Excellent.
Yeah. Anything else? Okay, cool. With that, I'm going to talk a little bit about the life cycle and the spread of HWA. So HWA does have a pretty complicated life cycle. In its native range, it even has a winged generation that we don't see here in North America. But the kind of main point that I want to take away from this slide is that there are 2 generations of HWA per year. The first is a long overwintering generation called the Sistens generation, and that generation kind of goes through the summer, fall, and into the winter, laying its eggs in the spring. And then there's a second, shorter spring generation known as the Pregridians generation, and that happens very quickly throughout the spring season. So by the end of winter, the first generation lays their eggs, and that spring generation emerges and begins its life cycle. Both life cycles kind of mirror each other. They both hatch from eggs into crawlers. They go through 4 nymphal instars where they are growing and getting bigger. They molt and they get a little bigger. They molt again, and so on. And that's when they start accumulating that wool. So you can see from this life cycle graphic, you're really going to see the wool kind of late fall through the spring season. So when HWA hatch, they hatch into their crawler stage. And the crawler stage is really important because it's their most mobile stage. So both generations of HWA hatch in the spring and early summer season. So that first generation is hatching by the end of March and into April. That second generation is hatching May and into June, early June. So they hatch from eggs into this crawler stage. And crawlers are when they're very mobile. They're really, really tiny, about 0.2 millimeters long. And so they can easily crawl onto a bird's wing or mammal fur and be moved from tree to tree. They can tumble through the canopy. They can cross onto new branches when canopies are touching each other. So crawlers are kind of the really easy stage to spread HWA. So I'd be careful April through June, just being aware that eggs and crawlers are going to be out there. So in the first generation, crawlers settle on twigs and become estivating nymphs. Estivation is a period of summer dormancy where HWA is not really growing or developing or feeding, but they have settled on the twig. Once crawlers settle on twigs, they don't move. They're there for the rest of their life. So in the in the summer, when that first generation settles on the twigs, they go into this period of estivation. They kind of look like little black sesame seeds surrounded by a thin halo of wool. And you can actually see them. So if you're out in the summer, it's helpful to bring maybe a magnifying glass or a hand lens with you in the field, because you can see these guys tucked in right near the base of needles. You can actually see them. I can see them with my naked eye, but it really helps to bring some magnification. So estivation is something that only cystins go through. The tree isn't moving that much material through its xylem during this time, so the HWA doesn't have much to feed on. So through July through October, you'll see these estivating nymphs waiting to begin feeding. Nymphs wake up in the fall, those cystins nymphs, the first generation. They feed, grow, and produce their characteristic white wool. So November through June is typically the best time of year to see that white wool. Once they've fed, adults lay their eggs, and they lay about 50 to 100 eggs per individual. So they are quite, they can reproduce quite rapidly and build up populations quickly. And this typically happens April through May. And as I mentioned before, that first generation lays their eggs starting in early April. We saw egg laying in early April this year in Ithaca. And then that second generation occurs quite quickly. They lay their eggs in the beginning of the summer. So I just want to reiterate again, you've got these 2 generations per year, with the kind of most visible time of year being that late fall through the spring. Wool does persist on trees, so I will say that even if you are kind of outside of that window, it's very possible to see wool from previous year's infestations. And the other thing I want to draw your attention to is that the cystins are the only life cycle, only generation that have that life cycle stage of estivation. During the spring season, when eggs hatch, crawlers settle, they immediately begin feeding. So what this looks like on the twig, especially because the tree doesn't necessarily have new growth on it all year round, new growth is kind of immediately colonized by that first generation. They go into their estivation, and then when they wake up in the fall and start feeding, that's when they start to get really fat and round. They put on that characteristic wool. But I want to draw your attention to this one special part of the life cycle that occurs on one section of twig, and that's when you've got these cystins adults, the first generation adults, and they lay their eggs. Once those eggs hatch, there's no new growth on the tree yet, and so the Progrediens generation, that second generation, settles on the same section of the twig as their mothers. So you've got this one section of twig where you're getting a lot of damage in one area because those cystins generation lays their eggs right there, and then all of the crawlers that hatch from those eggs are feeding from that same section of twig. And then once the tree pushes its new growth in the spring, that growth can be immediately colonized by the crawlers from the Progrediens eggs. So the next generation easily able to colonize all new growth on the tree. So HWA is a non-native. Not all non-natives are invasive. So I'm going to talk about the kind of 3 major reasons why HWA is so invasive. The first is that they can reproduce asexually. So it only takes that one individual to start a new infestation. So you just have one crawler landing on a twig, and they can reproduce quite quickly. And you can see if you're laying 50 to 100 eggs per individual, how quickly those populations are able to build up. The second is that there are 2 generations per year, so they're reproducing during those 2 different times of the year. Once again, building up populations quite rapidly. And we also have no native HWA predators here on the East Coast. So there's really nothing stopping HWA from getting out of control, and we've got no natural HWA population control. And so the most important thing to do now is manage those populations. Did I see that we have a question? No? Okay. So now I'm going to talk a little bit about HWA management. And we've got 2 ways that we manage HWA infestations currently available to us. The first one that I'm going to talk about today is chemical control. And then I'm also going to talk about biological control, which is what the lab that I work for is focusing on. So to talk about chemical control, I'm going to tell you first about the 2 products that we use here in New York to manage HWA populations. The first is a product called imidacloprid. The benefits of imidacloprid, excuse me, I'll start by talking a little, describing imidacloprid, and I'll talk about some of the ways that it's a really handy tool that we have. Imidacloprid is rather slow acting, so it takes about a year to become fully active in the canopy and knock back those populations. But it's long lasting. It can protect trees for up to 7 years. The other product we use is called dinotefuran. It is a fast acting product, so it takes about 3 weeks to become really active in the canopy, but it only has a short lifetime in the tree. It lasts for 1 season, 1 year in the tree. People who apply pesticide certified applicators can apply these 2 together as a tank mix. So you get the kind of benefits of dinotefuran, the fast actingness of it, and the benefits of the imidacloprid, how long it lasts in the tree. But for landowners, imidacloprid is the only product that's widely available for them. Landowners can apply imidacloprid privately as a soil drench. That's the only application method that's available to landowners. But certified applicators can apply it using a basal bark application, which is what you see in the photo behind the letters there. Stem injection, where it's injected directly into the tree. They can use release capsules that are buried in the soil. So there are many application methods that are available. I would recommend checking out our website's management section if you're interested in learning more about the application methods. Dinotefuran, however, is only available to certified applicators. So if you want that fast-acting effect, you'd have to hire a certified applicator that can apply it for you. So what you see behind you, that basal bark application is what I called it, that is what we consider our best management practice in New York. It is kind of the most economical. It's the cheapest but method that still reduces the off-target effects. So that is what happens during a basal bark application is that the product is applied to the bottom 0 to 5 feet of the tree. I usually use myself as a yardstick because I'm just like a touch over 5 feet tall. So it's applied to the bottom, just the bark of the tree. It's not sprayed all over the canopy. So it's a really great product or a really great application method because you can apply it in your water and you're not spraying it into the soil. So you're applying it directly to the tree itself. And that helps reduce the off-target effects that applying a product in a forest setting may have. So I want to talk a little bit more about imidacloprid specifically because it's the most common product that's used for HWA treatments. And it's also been widely studied, especially because you're applying something in a forest ecosystem. We want to reduce the risks to the rest of the ecosystem as much as possible. And I hopefully will be able to help you understand that applying this product in context is really important. So one of the important things to remember is that imidacloprid is a product that's designed specifically for insects. So it binds selectively in insect bodies, and that helps reduce the off-target impacts to mammals or birds or fish, other vertebrates. It has a lower risk to pollinators because hemlocks are wind pollinated. So people might know that imidacloprid is a neonicotinoid. Pesticides are typically kind of demonized in agricultural settings because they are a high risk to pollinator populations. But luckily, when we are applying to hemlocks, they're wind pollinated. So it actually poses a really low risk to pollinators when applying to treat HWA infestations. The last is because it lasts up to 7 years, it reduces the need for reapplication. So it's not something that you're doing every single season to control HWA. You're really only doing it once and you get that long-lasting effect. And ultimately, there are always going to be risks when applying products like this in an ecosystem, but there are also risks to not treating. And in the case of HWA, the major risk is that we'd lose all our hemlocks. So treatment really prevents the cascade of ecological effects that come from hemlock loss. So now I'm going to talk a little bit about biocontrol. So biological control in the case of HWA, we're using HWA predators from HWA's native range, bringing them here, releasing them in New York to manage populations, hopefully below the level of widespread hemlock mortality. So it is a long-term project. This is something that has taken years of research and will continue to take years of research into the future. But it is offering us a landscape scale solution. So you may have noticed that when I was talking about the chemical treatment of HWA, that you're applying it on a tree-by-tree basis, and treating individual trees is not always the best way to go about treatment. And biological control is a really great landscape scale solution because we can release predators in one stand of infested trees, and they are able to reproduce and spread throughout that landscape. The research is still in progress, and today I'm going to talk to you about the beetle species that we work with and silverfly species that we work with at Cornell. So here's a photo from our greenhouse at the Sarl Quarantine Facility at Cornell University. And I'm going to start to frame biocontrol by giving you an idea of the research timeline that usually is behind biological control. Because we're not just releasing predators into the environment without having studied them extensively first. And the first kind of major step to be able to release things safely in a new environment is research. So a lot of work has gone into way before releasing predators in a pest's new invasive range. To research those predators ahead of time. So that's kind of the first step in the biocontrol research timeline is identifying that pest species in its new invasive range and finding potential predators that may be able to manage that pest. The second thing you have to do is obtain permits. So once you have that research in place, you also need to get permission to release these things. So we're permitted at the national and the state level to release the predators that we do. And then you can release and continue research to improve the biocontrol techniques that you're using. So I'm going to talk a little bit about the species that our lab in particular works with. The first species I'm going to talk about is Laricobius nigrinus, a beetle species. And these beetles are from the Pacific Northwest. So as you may recall, HWA is native there. And these are a really abundant predator of HWA in that range. One thing that's really important about Laricobius beetles and why they make such a great biocontrol option for us is that they're a specialist feeder. So they only eat HWA. And that really helps us reduce the risk of releasing them in a new area where they, we don't want to release something that may eat many other things and disrupt a local ecosystem. So we've gone with a predator species that's a specialist feeder. They only eat HWA. The other thing about Laricobius that's great is that their life cycle syncs up with HWA. So they're a fall feeder. They're eating HWA when their first generation is growing and developing. So right after that estivation break, we like to release our Laricobius beetles, and they'll feed on HWA throughout the fall into the winter. And then in the spring, they lay their eggs. The eggs hatch into beetle larvae. The larvae drop into the soil and pupate, and then emerge as adults in the fall to begin feeding on HWA again. And we've released 9,705 of these beetles in New York. We've been releasing Laricobius. Our lab has been releasing Laricobius since 2008. So there they are in the lab feeding on HWA. The major things that we're working on with Laricobius beetles are collecting and releasing them across New York. So beetle prepupae and pupae are collected from the native range in the Pacific Northwest. Once they emerge, we release them here in New York. And we're also monitoring for beetle establishment at previous release sites. This is really important for making sure we know where we are having biocontrol success. But also we just want to know if it's working in general, if those populations are able to reproduce. And although we're not seeing any widespread HWA population control yet, we have found beetles at previous sites. I'll talk a little bit more about that later. The other species that we work with are Leucopus silverflies. So we work with two closely related species of silverfly, Leucopus argenticalis and Leucopus pinnaperta. And silverflies are extremely abundant, maybe the most abundant predator of HWA. These are also Pacific Northwest natives, so we are collecting them from the US and Canadian Pacific Northwest. And this species eats HWA eggs. So they're spring feeders, not really competing with our Laricobius predators, but in fact providing an HWA population control during another part of the HWA life cycle. So they're eating the larvae of these flies are eating the eggs of HWA. And we've released about 10,000 of these flies throughout New York. We've been releasing them since 2015, and the New York State Hemlock Initiative was the first lab to release silver flies for HWA control on the East Coast. So this is how we conduct our silverfly releases. We often release them in bags. So with silverflies, we are doing a lot of different research on silverflies, but kind of our main releasing program is focused on collecting flies from the Pacific Northwest to release them across New York. That photo from earlier showing all that foliage in a box, that's how we collect our flies. We're also working on developing lab rearing techniques. So we're seeing how well we can rear flies in the lab to release them. It is a little complicated because with specialist feeders, you have to provide them with the food that they need year round, or else use laboratory climate controls to alter their life cycle so that we're able to feed them and keep them for a long time in the lab. We're identifying genetic microsatellites, so we're looking into ways that we can find flies after we've released them into the field and be able to tell them apart. And we're investigating niche separation. This is a new project that we've started this year, looking into when the differences in the life cycle between the two different predator species that we work with. And we're hoping that if we can do this correctly, that we can release the predator species together. And so Leucopus argenticalis gets released all together with other argenticalis individuals. And the Leucopus pinnaperta get released with all other pinnaperta individuals. Our preliminary data shows that there's When our predators are emerging, so when we collect them for release, the Argenticolous are emerging first, and then the Pinniperta are emerging later, and then there's a second wave of the Leucopus argenticolous. And so this will help us better time our biocontrol releases during the spring season. So a few kind of measures of success that we've had.
Hey, Charlotte, just quickly on this, someone was asking, what's the size ratio? You know, how big is a Laricobius beetle as compared to the Adelgid? I know you had a slide that showed it a little bit, but just a sense of, yeah, if we could go back to that. There you go. So we can see there's the beetle, and then the Adelgids are not visible to the naked eye. Is that right? Or they're the little sesame seeds?
They are, so Adelgids when they're adults are about 1 millimeter long. So these beetles are a little bit bigger, definitely. But the HWA, you can see some of the white woolly masses there. They are visible. So although the beetles are a little larger, the beetles are quite tiny. I mean, they're you can see just how large the needles of that hemlock look. And the beetles are less than 1 inch long. So you can imagine the beetle isn't even half of that. So they're really tiny.
Excellent. Thanks for doing that and going back to that slide.
When we've reared them in the lab, we have to pick them up using paintbrushes, little like kids craft paintbrushes, because they're just really tiny and delicate. So we want to make sure that we don't smush them when we pick them up. And the flies aren't much bigger. They're about the same, around the same size as the, they're a little over 1 millimeter long. So if the adult HWA are about 1 millimeter, they're only slightly bigger. And I actually have another photo coming up. There will be a photo of the fly larvae, and you'll be able to see the larvae with HWA eggs and see the size difference there. So, a little bit about our biocontrol success. Last year, we were able to release well over 6,000 flies. So that was a really, really exciting year for us. We had a bumper crop of flies, and we were able to release a lot of flies last year. And we also found that we have confirmed establishment of the Laricobius beetles at 5 release sites. And we have I think we have 21 release sites for Laricobius, and finding them at 5 was really encouraging. So this next year, hopefully we will improve our predator establishment techniques, try to find some more predators, and hopefully we'll find evidence of predators at other sites. So I am going to talk a little bit more about the biocontrol research, but I'm also going to mention and kind of our future directions, things that we're going to look into. But I want to talk a little bit about our HWA research as well, because we are still learning more things about HWA. One of the things that is really important to our work is observing HWA phenology. And to do this, we work with the National Phenology Network with a program called Nature's Notebook. And so we have citizen scientists who help us track the timing of HWA's major life cycle stages throughout New York, because it can differ based on the climate and other factors. So temperatures is one that we found. Growing degree days tends to be kind of one of the major ways that phenology can differ across New York. So we do this because it's really important to release our predators when the right life cycle stages of HWA are around. So we know that beetles need to eat live, growing, developing HWA. So we want to release them after estivation break. So after they break out of their summer dormancy period. So that's kind of one major life cycle stage that we track. And we also need to release flies when their larvae can eat HWA eggs. So another major life cycle stage that we are looking at the timing of is egg laying. We really want to know when HWA begins laying eggs so that there's food available for our predators when we release them. We monitor HWA winter mortality. So we're comparing warmer and cooler sites in New York to see the proportion of the population that are surviving major cold events. So when we get those really cold snaps in New York, we like to compare between the colder site and the warmer site, air quotes there, colder and warmer sites. In New York, we like to see what the difference is in HWA surviving populations. So in some cases, a really cold night can result in over 90% of the HWA at that site dying. So comparing those two sites, we're trying to learn more about how HWA responds to cold weather. We're looking for new biocontrol release sites. We actually have one staff member who's now full-time working in the Catskills to help us identify HWA infestations, find them, and look for great release sites. And we do look for really specific criteria because we can't just release where any infestation is. We have to release in places where the trees are going to survive long enough. So it has to be relatively healthy trees, but with high population density of HWA, because we want to have a lot of food available for our predators. So we really need to, it can't just be every single HWA infestation that we want to release bugs at. We really want to find those sites where our predators are going to have the best chance to survive and reproduce. And this year we started using imaging techniques to observe tree damage. So looking really, really closely at the twig and finding out exactly the physiological damage that's occurring when HWA is inserting that needle-like mouthpart into the twig. So this is the photo I was talking about. This is a Leucopus larva, and it's feeding on the eggs of HWA. So all those tiny little yellowish eggs, yellowish goldish eggs. You can see orange eggs just north of that Leucopus. Those are the HWA eggs. So they're feeding on those eggs. So one of the kind of things that we're really, really excited about this year is using eDNA, or environmental DNA, to confirm predator establishment in the field. So we're developing techniques to do this now. So we're hoping to deploy our DNA collectors soon. But we're really hoping that what we can do is we'll be able to use eDNA to tell us if predators have been there, so that we're not just seeing them with our own eyes. Especially with the flies, it's very impossible to track them once we've released them in the field. And with beetles, the techniques that we're using now are not as fine-grained as we'd like. So we're hoping that using this technique, we'll be able to spot predators in the field very easily. We're also working with a graduate student to compare hydrological dynamics using both modeling and field data at sites with healthy and HWA-infested hemlocks. So we're looking into how tree health plays a role in hydrological dynamics in different sites, specifically in the Catskills, but also across the East Coast. And preliminary data, recently published data, says that, or showed that at sites that have HWA-infested trees, so trees that are in poorer health, actually had higher stream flashiness after precipitation events, which would cause greater, which would result in a greater risk of flooding. So we are looking into the kind of larger impacts of HWA infestations over long timescales with this, seeing how it plays into the dynamics of full watersheds. Any questions about our research? Okay, I'm going to move on and talk about the ways that everybody can get involved with Hemlock woolly adelgid in their region. So the first is to know what's going on in your local area, and your PRISM, or your Partnership for Regional Invasive Species Management, is a really great way to do this. So I'm going to shout out again to our hosts, the Adirondack PRISM, APIP. They are responsible for all sorts of HWA related things, but also all the invasives in the Adirondacks are covered by this group. So one of the ways to get involved with HWA or any invasive is to check out your local PRISM, because they're going to have all the answers for every invasive in your region. So I just want to do a shameless plug for the APIP Prism here. Moving on, landowners, as I mentioned, play a really, really outsized role in forest stewardship. So if you're a landowner and you're worried about HWA, what you can do to help is first find your hemlock trees. Know where you have hemlocks on your property and how much of your property is hemlock. That can be a really critical step, especially if you find hemlock woolly adelgid infestations. You'll be able to know exactly where the at-risk areas on your property are. And that will help you prioritize management down the road if you find yourself with HWA. Then you'll want to survey for HWA on your property once you find those hemlock trees. If you don't find any, we recommend going back once or twice per year. We do have a volunteer program specifically aimed at landowners that encourages regular surveying. So you can look that up on our website if you're interested. And the other thing to do if you feel like you would like to is to treat infestations. But as I mentioned, there is a risk to not treating as well. Especially in the Adirondacks, it's really important to conserve those hemlock resources because you guys are the densest hemlock region in New York. You guys have the densest stands, really healthy hemlocks, everywhere from new to old growth. So hemlocks are a really key feature. So landowners can really help by treating any infestations they find. If you're a member of the community, maybe you don't have any hemlocks on your property, you can go to areas with hemlock trees, survey for HWA there, and then it's really important to report your HWA infestations. If it's not reported, no land manager can deal with it. So it's absolutely critical, and I'll reiterate it again, in the Adirondacks especially, it's very critical to report those infestations early so that treatment and management techniques can come in and help manage those populations. So a few survey tips if you're going out to look for HWA. The first is to go out during that best time of year to see HWA. If you want to see those white woolly masses, it's best to go kind of November through May. The next is to consider tree health. So if you're seeing a hemlock tree that's maybe got some dead branches, maybe has a pale grayish appearance, is losing needles, you'll want to look a little closer at that hemlock tree. It's really helpful to look for branches that are on the ground and see if there are infestations. This is especially helpful in areas where you can't reach any branches. If the canopy is really high, just look down on the ground and then you might be able to find some branches and see what's going on up in the canopy. Surveying areas where there are branches in reach is helpful because then you can get up close and personal with that HWA. Looking at the undersides of twigs and branches is another great way to find HWA. They do tend to clump on the underside of twigs. So kind of flipping up branches when you're looking is helpful. And check branches on all sides of the trees. As I mentioned, even that one individual can start an infestation. So if you look on one side of a tree that hasn't had that individual arrive yet, but the other half of the tree is all infested, you could miss an infestation, get a false negative if you're only checking that one branch. So it's really helpful to check branches all around the tree. If you're a boater and an angler, I like to throw this in, keep an eye out when you're on the water. Hemlocks, will start to lose their needles when they have infestations, or they'll start to die back, have this pale grayish appearance. So keep an eye out for those ghost trees where they start to look really gray and unhealthy. The other thing to look for is the lack of new bright green buds in the late spring to early summer. So in the late May to early June, hemlocks start to get this, their new growth. And it comes in and you can see it's really, really bright green, like almost tennis ball green. So those new buds are really easy to see from the water. And if you see a hemlock tree and that doesn't really have that new foliage on it, that could be an indication that the tree is having a hard time pushing new growth. So I want to just let everybody know to keep an eye out for that. The next thing to do after you're done surveying is definitely report HWA infestations. One of the easiest ways to do this is by using the New York IMAP Invasives mobile phone app. So it's really easy. You take photos of infestations on your cell phone, you upload it on, and then that contributes to a statewide map. So the map I showed you of New York earlier and the spread of HWA, many of those infestations were reported by citizen scientists using IMAP invasive. So there's their website. I would definitely recommend checking it out because it's the easiest way to report infestations to the state and to us. We actually get every single HWA report from IMAP. So everybody involved in HWA will get that report. The other thing that's cool about IMAP invasives is that you can report that you didn't find an HWA infestation, and that really helps us know where our hemlock resources are in New York and where infestations haven't reached yet and where's been surveyed. So if people are looking and saying, oh, it doesn't look like that area has been surveyed, it might have been. So you can report, oh, I did survey, I didn't find anything there. And that really helps people know where people have already been looking for HWA and where great hemlock resources are. So I highly recommend using IMAP invasives. It's a great tool. The other thing is we have a HWA reporting tool on the New York State Hemlock Initiative website. And if you're looking for a way that's not online, you can call the DEC. There's their number there, and you can report forest pest infestations using the phone. So a few final thoughts before we leave, things that I want to talk about. The first is that we really can't rely on winter temperatures. There's a lot of publications out there that will say winter temperatures knock back HWA. But unfortunately, what we've seen is that there is no natural population control that will keep trees from dying. We've lost millions and millions and millions of hemlock trees on the East Coast. And so it has been shown that winter temperatures are not knocking back populations and keeping trees from dying. So we really can't just rely on cold winter temperatures to keep our trees safe. The next is that HWA surveys and treatments are critical. Chemical control is really the only tool that we have now that's a sure thing for saving that hemlock genetic diversity in the short term. So biocontrol research is continuing, but right now treatment is really the only way that we have to keep trees on the landscape and save that hemlock genetic diversity. And HWA surveys are the way that we can manage because without knowing where populations are, we can't do anything about it. And the next is that biocontrol is really the adapt and move forward move for us. This is the way that we have to adapt to HWA populations because it's here. It's been in New York since the 1980s and it's established here. So biocontrol is really the long-term landscape scale solution. And so between the chemical treatment and biological control treatment, we really hope that we'll be able to manage populations below widespread hemlock mortality. That's the way we're going to do it. So I just want to say thank you to everyone again. There's our website, so definitely visit us online. All the information I told you and more is on our website. We also have Facebook and Instagram, so you can stay in touch with us. And we also recommend, if you are interested in learning more, you have questions, please email us. This email address goes to 4 people in the New York State Hemlock Initiative organization, so somebody will answer you. And we have a lot of knowledgeable staff that are ready to help you and share all sorts of information about hemlock trees and HWA with you. So thank you all, and I'd love to take any questions you may have.
Great, Charlotte. Thank you as well. We do have a few questions. I want to remind people that we are recording the webinar and it will go on our Adirondack Invasives website once we get the recording finished. I know some people may have to drop off the call, but I do have a few questions that are coming in for you, Charlotte. And I just want to reiterate what Charlotte, we've got a lot of folks on the call today who are active and out and about in the Adirondack and eyes and ears and boots on the ground. So if you think you've seen Hemlock woolly adelgid, take a photo of it. If you can get coordinates of where you are, that's wonderful. If you can upload that to IMAP invasives, even better. But if you can't do those things or you can just snap a photo of it, try and remember where you are. Figure out if you can, tie a bandana out there or some way that you could get back to that site. Let the Hemlock Woolly Adelgid Initiative know about it, or let DEC know about it, or you can contact us at APIP, and we'll try and get out there and see if we can verify that that's out there or not. So I can't thank everybody on the call enough for being the eyes and ears out there on the ground. We'd also love to get your feedback. I put my email in there, but if you can let us know what you liked about today's webinar, what we could do better in a future webinar, or what other topics you'd like to hear about, I'd also appreciate that feedback too. Okay, Charlotte, I've got a couple of questions that have come in for you.
Sure.
We had one earlier on, which was somebody who said, I've got some important trees in my backyard. There might be some in local cemeteries. Is there any kind of nutrients we can apply to those trees? This is different from the imidacloprid, but is there any kind of nutrients, anything we can do to make them healthier so they might not be as subject to the Adelgid?
That's a great question. We haven't actually, so I guess in a roundabout, I don't actually know if there's really nothing that's going to prevent infestations that we know of. You can fertilize them to keep them as healthy as possible, but it seems that there really isn't any resistance. There is a lab at the University of Rhode Island that's looking into the question of native resistance to Hemlock Woolly Adelgid. But we haven't seen any documented cases on the East Coast that, so pretty much all hemlock trees are susceptible from what we've seen. That may change, research is continuing in this area, but as far as I know, there really isn't a nutrient or anything that's going to protect the trees.
Okay, well, I have another follow-up question that's pretty similar along that line. So we've got some folks on the line that work for Department of Transportation, and they happen to be removing a number of hemlocks, and they're going to be replanting hemlocks in those areas. Do you think it makes sense to actually preventatively treat those new hemlocks with imidacloprid just to prevent bringing in HWA? What should you do if you're going to take new nursery stock and put it into a new place in New York State?
Well, definitely check that nursery stock for infestations before you plant. That's huge. So that's kind of the most important thing that you can do. We don't recommend that people preventively treat. That's something that's left at the agencies, though. I know that in the case of, say, the Adirondack infestation that was found on Prospect Mountain in Warren County, they did treat a buffer zone. So they preventatively treated it, or preemptively treated, a group of trees around the known infested trees as a preventative measure. But especially if you're a landowner, I don't recommend that kind of treatment, because HWA is spotty. I mean, even in that infestation slide I showed you, you could see places where they're within the infested zone, largely infested zone, that there were spots where HWA hadn't been founded. So I guess I would say I don't recommend preventatively treating. But since it's DOT and not a private landowner, I would recommend emailing that email address at the bottom and asking, because somebody with a little more management experience can give you an answer that might be more detailed than I can. But we don't recommend preventatively treating, because typically you can't be sure if an infestation is going to find your tree. There are a lot of situations where kind of an isolated tree with not a lot of other hemlocks around it never gets an HWA infestation. It's a really hard to predict insect. So even two trees next to each other, one may be infested, one may not be. So yeah, I don't recommend treating unless you're absolutely sure you have an infestation there. But part of that is just for ecosystem health and cost.
Yeah, I think that's really, no, that's helpful, Charlotte. And I think also, so I love the fact that DOT should be contacting you for more information on treatment. And also, I think there's a good reminder out there that if you are bringing in, if you're a private landowner or you're doing a little bit of landscaping and you're bringing in new hemlock trees, you're buying it through a nursery, check that hemlock very carefully before you plant it. We have seen it transported in New York State through nursery stock. So can't emphasize enough checking it before you plant.
Yeah, and I will say, like, New York works very, very hard to reduce that risk that it would spread through infested nursery stock. We do have horticultural inspectors that go to nurseries and check for infested stock. So that being said, you know, it can slip through the cracks. Just one little tiny HWA can start a whole new infestation on a tree. So even though we do have a lot of people on the front lines, it's really hard to detect every single tiny invasive individual on those trees. So it's really important that people buying those trees or other plants are checking just in case.
That's great. And we had one more research question, and this was about the biocontrol flies. So if the flies are feeding on the spring eggs, what are the flies doing the rest of the year?
So we actually are looking into this now. We have an overwintering experiment going on. So we released some flies in those mesh bags or bags of pupae. So we're trying to see if the flies overwinter as pupal in their pupal stage. And we did actually see flies emerge from our overwintered pupae. So what we're seeing is that there are probably life cycle stages where the flies either arrest their development or are in a prolonged period of pupation and are able to ride out. I mean, it's happening in their native range, so we know that it is possible, but we're really looking closely this year at the fly phenology and trying to learn as much as we can about how their life cycle occurs and how their life cycle might change in New York versus in the Pacific Northwest.
This is Tom Green. I'm the one who asked that question. I'm from the Hemlock Restoration Initiative in North Carolina, actually. I was glad to join you guys today. Where do they pupate? I might have missed, you just said that.
Oh,
is there any idea where they pupate?
Yeah, they pupate on the needles. So
on the branch. Okay, great.
Yeah, they pupate right on the tree.
Great, thank you.
I think that might be the end of our questions here. Hopefully I've gotten all of your questions answered. And once again, please let me know what you liked about the webinar, what we can do better, or if you have other topics you'd like to learn about. I want to thank Charlotte for her time today. And I want to thank all of you for, again, spending your Friday morning with us. So thank you very much. And we will post the webinar recording on our website. Have a great weekend, everybody.
Thanks, everybody.